To understand what complementary DNA or cDNA is, we really need to start with a general discussion of the different types of nucleic acids that naturally occur within organisms. So, of course, we've heard about genomic DNA. From those instructions, we generate transcripts, which are messenger RNA, and then from those transcripts, we produce proteins via the ribosome. And the proteins are the real functional elements inside all of the various cells that an organism has that create phenotype within those cells. For example, the brain, the cells within the brain, both the neurons and support cells, their job collectively is to coordinate behaviors in humans and to regulate many different processes around the body. Whereas the liver cells here are involved in getting rid of toxins and regulating various aspects of body function as well. The phenotype of the brain and the phenotype of the liver cells are very, very different. And they're different because different proteins are expressed in those two different tissues. The way that the genomes within these liver cells activates these proteins is mediated through the selective transcription of mRNAs. Thus, there are very different mRNAs and proteins in the liver than there are the brain. Okay, and that's because there are very different intended functions for those cell types. However, the genomic DNA in those cells is identical. Okay, so these blue arrows here indicate expression. Expression is highly regulated from cell type to cell type within an organism, be it muscle, the cells that produce bones, digestive cells. There's over 200 different cell types in the human body. Let's take, for example, 1,000 neurons from the brain. So let's say we've isolated 1,000 of those cells. And let's take 1,000 hepatocytes from the liver. Okay, so we're looking at 1,000 of those cells. If we were to isolate the genomic DNA from each of those two samples, what differences would there be? If we were to take all of the messenger RNA from those two samples, what differences would there be there? And lastly, if we looked at the total protein content between those 1,000 neurons and those 1,000 hepatocytes, what kinds of differences might we see there? Starting with the genomic DNA from these two samples, you can see that there's absolutely no difference in the genomic DNA content between 1,000 liver cells and 1,000 brain cells. Each one of those genomes has a single, in this example, human chromosome 7. Okay, and if human chromosome 7 happens to have 3 particular genes on it, neuropeptide A, liver enzyme B, and ribosomal subunit C, so 3 generic genes that I'm making up, there will be 2,000 copies of each of those 3 genes in 1,000 neurons, and there will be 2,000 copies of each of those 3 genes in 1,000 liver cells. And the reason there's 2,000 is because these are diploid cells we're talking about, and each one has 2 chromosome 7s in it. Okay, so there should be no variation between the gene copy number for genomic DNA specifically. Now, if we contrast the messenger RNA in those samples to genomic DNA, we'll see that in neuron cells, you may have lots and lots of copies of neuropeptide A being made from its genetic locus on chromosome 7 there. And that's because neuron cells are going to be potentially using that particular transcript to build a lot of the neuropeptide A protein. Whereas the liver enzyme B maybe has no production whatsoever, because liver enzymes typically aren't used for any functional purpose inside a neural cell in a human. Okay? And a third situation with the ribosomal subunit C, where perhaps there is some expression of that going on because every cell in the human body, or I should say most every cell, needs ribosomal subunits for basic translational purposes. Now in our 1,000 liver cell sample over on the right, you've got a different situation where there's probably 0 expression of a neuropeptide in that situation. You've probably got lots and lots of expression of mRNAs corresponding to the liver enzyme that needs to be built there. And again, you see some expression of ribosomal subunit C because translation is taking place in those cells as well. What the graphical display of gene expression is going to be in these two samples is very high expression of neuropeptide A on the left case, very low to zero expression of neuropeptide A on the right here in our liver cells. Contrast that with liver enzyme B that doesn't express very highly in neurons and is expressed very highly in hepatocytes or liver cells. And then pretty even expression between the two tissues and the mRNAs corresponding to ribosomal subunit C, since that's a general process of most, if not all, cells in the human body. Protein content, of course, would be very similar to these levels in those two tissues, assuming that the protein to mRNA ratio is relatively constant. When I go to isolate genomic DNA from these two different samples, I'm going to get essentially the same thing in each sample. I'm going to get a ton of double-stranded genomic DNA from those 1,000 neurons, and I'm going to get an equivalent amount of double-stranded DNA from these 1,000 liver cells over here. Every human gene known in the human genome will be represented in equal copy number in each of these two samples. Double-stranded DNA is very stable, so it's actually ready to go in this form for things like PCR. Messenger RNA, on the other hand, is single-stranded, as we know, and has that extra hydroxyl group, making it highly reactive. And so it's not as useful for direct applications. There's a way around this, though, and that's to create a complementary DNA library, or cDNA. And in that way, we can create double-stranded copies of the messenger RNA transcripts while preserving the quantitative aspects of each sample that we're working with. So if there are tons and tons of copies of gene A in our 1,000 neuron sample, there will be corresponding tons and tons of copies of the complementary DNA after we've processed the mRNA from that sample. And if there are no copies of gene A in our 1,000 hepatocytes mRNA, then there will be no mRNA in the first place to build our cDNA library off of, and those cDNAs will not be represented in that particular library. So let me quickly walk you through how a standard cDNA library is constructed. Let's take one messenger RNA, okay, and of course we have, if we're looking at human cells, we have the 7-methyl guanylate cap over here, okay, and of course we have a poly A tail for most coding transcripts. So this is a protein coding messenger RNA that's destined to leave the nucleus and go find a ribosome. It has this nice poly A tail to help signal and move it around, but also for stability to not mess up this 3' end. This is a really convenient feature when trying to make cDNA because we always need a primer to create new nucleotide sequences. In this case, we're not going to use DNA polymerase. We're actually going to use something called a reverse transcriptase enzyme. That reverse transcriptase does need a 3' hydroxyl group to build new nucleotides, just like the other enzymes we've looked at this semester. And so to provide that, we can put into this reaction a poly DT primer, okay, that's going to be made up of all T's, and that will hydrogen bond with that poly A tail, providing a 3' hydroxyl group at that leading edge there, coming back this direction to copy my messenger RNA. At this point in the reaction, all of the mRNAs that were there to begin with have been copied and amplified, but they're being copied into DNA at a rate that's consistent with their original levels. So if there's a lot of mRNA A, then there will be a lot of corresponding DNA made from mRNA. If there's not a lot of mRNA-B, there will be a consistently small amount of cDNA made from mRNA-B. The next step in the reaction is to get rid of the mRNA, and typically this is done with heat or sodium hydroxide, some harsh chemical. And since mRNA is less stable, it just sort of breaks down. We can then use random hexamers to prime a standard DNA polymerase and build the original mRNA sequence back in DNA form all the way to the end, such that we have a nice stable double-stranded copy of the original mRNA that we can now do things like downstream PCR or quantitative PCR on, which is very useful in molecular biology.